This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas.
Welcome to the Deep Dive. You gave us the sources, we wrestled with the data, and now we're here to give you the most critical insights from the cutting edge of science.
Today, we are wading into a really complex, evolving, and frankly, a pretty worrying story about the highly pathogenic avian influenza H5N1 virus.
That's right. And if you thought you were well informed about the H5N1 episodic that started tearing across North America back in 2021, you might want to prepare for a major update.
Because the rules of the game have fundamentally changed.
Absolutely. Our deep dive today centers specifically on a new player, a genotype called D1.1.
And this particular variant has, it's just completely altered the landscape of the H5N1 episodic in North America since it emerged in mid-2024. This isn't just another small mutation.
No, this is what the researchers are calling a major evolutionary leap forward for the virus.
Right.
So let's unpack this right away, because the headline finding here is dramatic. Since H5N1 first showed up on our continent in 2021, we've been tracking, high mortality in wild birds and domestic poultry.
A huge problem for years now.
But this D1.1 genotype, which appeared last year, introduced what the researchers call a major shift.
And they don't use that term lightly.
Not at all. It's proven to be faster spreading. It targets the broadest range of hosts we have ever seen. And crucially, it was responsible for that infamous host switch to dairy cattle in new states.
And, tragically, caused 2 documented human fatalities by early 2025.
The stakes are incredibly high, and the scale of the scientific investigation needed to track this is it's immense.
It really is. The researchers, and we're talking about a team led by Alvin Crespo-Bellido, Nydia S. Travaun, and Martha Thurst-Nelson, they use these advanced forensic methods called Bayesian phylogenetic analysis.
Okay, so it sounds complicated, but what does that actually let them do?
It lets them look at a truly massive data set. In this case, over 17,500 H5N1 genome sequences from all over the world. Yeah. And their mission wasn't just to catalog the cases. It was to definitively establish when D1.1 emerged, where it came from, and, maybe most importantly, how fast it was able to evolve and spread compared to everything else that came before it.
So it's the closest thing we have to a viral CSI team.
That's a great way to put it. They're tracking the complete history, the movement, the entire family tree of this organism.
So for those of you who follow these topics closely, let's start with the baseline. We need to set the scene and really understand the H5N1 environment in North America before D1.1 showed up and started breaking all the rules.
Okay, so the original story begins with what scientists call H5N1 clade 2.3.4.4b.
Right, the highly pathogenic strain.
Exactly. It had been circulating widely in Eurasia since about 2016. Its grand entrance into the Western Hemisphere happened in November 2021.
But it was carried by migrating wild birds from Europe, correct?
That's right. They used the Atlantic flyway and brought the virus to Newfoundland in Canada, and that's what established the initial North American foothold.
And as soon as it landed, the clock started ticking on what we call genomic reassortment.
Yes, and this is the main evolutionary engine for influenza viruses. We've talked about it before, but it is so crucial to revisit here because it dictates everything that follows.
So just as a refresher for everyone, can you break that down?
Absolutely. Think of the influenza virus genome not as a single long strand of RNA, but as an 8-piece jigsaw puzzle.
Okay.
These 8 pieces, or genome segments, they encode all the necessary components for the virus. So when a single host, usually a wild bird like a duck or a goose, is co-infected at the same time with 2 different viruses.
Like the foreign, highly pathogenic H5N1 and a native North American low pathogenicity avian influenza, or LPAI, virus.
Exactly. When they're both in the same cell, those 8 puzzle pieces from both strains can get all mixed up and then repackaged.
So if a new virus particle gets, say, 4 segments from the Eurasian strain and 4 from the North American strain, you essentially get a brand new hybrid virus.
You do. And this is so much more dramatic than a simple point mutation, which is just like changing a single letter in the genetic code.
This is like swapping out entire chapters of the instruction manual.
Precisely. This reassortment creates entirely new genotypes with novel combinations of genes, and that gives the virus massive evolutionary options very, very quickly. It's why the H5N1 situation in the Americas has been so dynamic and so difficult to track.
And to manage all this complexity, the sources rely heavily on a specific naming system, a nomenclature.
Yes, the GenoFlu system, which was developed by the U.S. Department of Agriculture for tracking.
Can you just quickly break down what A, B, C, and D mean in this context? Because we're
Hmm.
going to be using those terms a lot.
Sure, it's actually a pretty essential tool for describing this vast array of new strains. The original non-reassorted Eurasian viruses, the direct imports, are categorized as A genotypes.
Okay, so A is for the original ancestor.
Right. Now, once those A viruses start swapping some of their internal segments with the North American LPAI viruses, the new mixed breed viruses are categorized as BC, or in the case of our focus today, D genotypes.
So it's basically a shorthand for identifying how much of the virus is foreign and how much is locally acquired.
That's it, exactly.
So let's trace those initial pathways. From 2021 into 2022, North America is huge, and the virus used the migratory paths, the Atlantic, Mississippi, Central, and Pacific flyways to get around.
And the initial success story, for the virus anyway, belonged to the Atlantic flyway, the European origin lineage, which was genotype A2, and which then diversified into A1, that established the most robust foothold.
And this is a really critical point in the
It is,
paper.
it is. The Atlantic introduction was the only one on the East Coast that managed to sustain long-term transmission and consistently fuel that reassortment process.
So that's what gave rise to the B and C genotypes that really dominated our landscape in 2022 and 2023. But the Pacific Flyway, the West Coast, that saw a very different pattern. They were getting viruses from Asia.
They were, and this distinction is absolutely crucial. The Pacific Flyway experienced repeated introductions of the A3 genotype, originating from Asia.
And not just once.
No, the study documents at least 6 distinct introduction events. However, most of those A3 introductions just failed to launch. They fizzled out really quickly and didn't establish sustained transmission.
They were basically evolutionary dead ends.
For the most part, yes, they were dead ends, except for one.
There's always an exception.
Always. The sources indicate that the third A3 introduction was the one. That was the crucial lineage that managed to survive, to spread, and to eventually initiate its own reassortment process.
And that lineage is the direct ancestor of our problem child.
It is the direct ancestor of the D genotypes, including our main topic, D1.1. And it just shows how persistence, even after multiple failures, can eventually lead to a major evolutionary event.
So before D1.1 emerges in 2024, when the virus was actively swapping these puzzle pieces, what were the go-to swaps? Which parts were most commonly exchanged with the native North American viruses?
Before mid-2024, the swaps overwhelmingly involved the internal machinery of the virus.
So not the parts the immune system sees on the outside.
Exactly. We're talking about the viral ribonucleoprotein complex, or VRNP.
Okay.
This complex is made of 4 key internal segments. The 3 polymerase segments, PB2, PB1, and PA, and the NP, or nucleoprotein segment.
And these internal genes, they basically dictate the speed and efficiency of how the virus replicates inside a host cell.
That's a perfect way to put it. Swapping these segments often allows the foreign H5N1 to suddenly replicate much, much better in North American birds. It's like getting a new engine.
This detail is often overlooked, but you're saying it's the critical baseline for understanding the danger of D1.1.
It is, because the 2 surface segments, the ones our immune systems recognize, hemagglutinin, the H5, and neuraminidase, the N1, they were fixed.
They were always the Eurasian version.
That is the absolute non-negotiable fact of the 2021 to 2024 North American epizootic. Until D1.1 emerged in October of 2024, 100% of the H5N1 viruses circulating in the Americas had retained that original Eurasian N1 segment.
So all the visible evolutionary diversity, all those B and C genotypes that caused so much damage, that was all happening internally.
All under the hood.
And we had plenty of drama from those internal reassortants already. Before we move on to the D series, let's just take a moment to contextualize the impact of those earlier genotypes. They really established the pathways for cross-species transmission.
They absolutely did. You can think of them as proof-of-concept viruses. For instance, we had genotype B3.2.
Right, that's one that caused those truly devastating mass mortality events in South America.
Yes, wiping out tens of thousands of sea lions and other marine mammals in places like Peru and Chile. That lineage demonstrated H5N1's new ecological danger to marine ecosystems. It was a huge wake-up call.
Then we saw B3.6.
B3.6 was notable because it was one of the first to show a domestic mammalian spillover, infecting goats in Minnesota in March of 2024.
So that was an early warning sign that these internal swaps were expanding the host range beyond just birds and the usual poultry.
A very clear warning sign. And then, of course, there was the incredibly significant B3.13.
Which is perhaps the most famous precursor.
It is. B3.13 was the genotype responsible for causing that massive multi-state outbreak in U.S. dairy cattle during 2024 and 2025.
An outbreak that, according to the sources, was still actively spreading at the time this paper was published in December 2025.
And B3.13 was a pure internal reassortant. It showed that H5N1 could sustain itself not just in birds or poultry, but in these large-scale domestic mammal operations, which just dramatically changed the economic and public health risk.
So we had 3 years of internal tweaking, host jumps, ecological devastation, all following one rule.
Yeah.
Keep the Eurasian N1 surface protein.
Wait.
But now we turn to genotype D1.1, the turning point where the rules change completely.
This is where the story takes a very sharp turn.
So genotype D1.1 didn't emerge in that familiar Atlantic flyway where most of the B and C genotypes came from. It came from the Pacific. What did the phylogenetic tracking tell us about its exact origin story?
Well, the advanced analysis the research team used allowed them to pinpoint its time to the most recent common ancestor, or TMRCA, with surprising precision.
Okay, so when did it first appear?
In a really tight window in mid-2024, specifically between July 19th and August 9th.
So that means D1.1 was circulating totally unrecognized in the wild bird population for several months before anyone actually detected it.
Exactly, it was moving silently. And its first official detection told the researchers exactly where to look for its ancestor.
Where was that first detection?
It was in western Alaska on October 6, 2024, in a wild duck and northern pintail.
Which confirms its specific lineage. So it came from that sustained third A3 introduction from Asia that we talked about.
Yes. The narrative is crystal clear. This was a West Coast evolutionary event that started moving inward and eastward, which is contrary to the dominant east-to-west spread of the previous years.
Now, let's get to the genomic core of why D1.1 is so significant. What was its unique genomic composition that set it apart from every single previous North American reassortant?
D1.1 completed a perfect 4-in-4 swap. It kept 4 segments from its original Eurasian A3 ancestor, that's the HA, or H5, plus PB1, MP, and NS, but it acquired 4 new segments from North American LPAI.
And what were those for?
Those were PB2, PANP, and the crucial game-changing segment, NA, the N1 neuraminidase segment.
The neuraminidase segment. That is a massive fundamental change.
Huge.
Because that dictates how our immune system and existing vaccines see the virus. This is an antigenic shift.
It is the definition of an antigenic shift at the reassortment level. This makes D1.1 the first major H5N1 reassortant to acquire a new surface protein gene, a new neuraminidase, or N1, from a North American LPAI lineage.
The virus essentially changed its ID tag.
That's a great analogy. If the old H5N1 was wearing a European uniform, D1.1 suddenly put on a North American uniform, and that could allow it to potentially evade any existing immune responses that might have been building up against that older Eurasian N1 version.
And the data shows just how advantageous that shift was in the wild bird population. This was not a slow, gentle evolutionary push, was it?
Absolutely not. The new D1.1 lineage rapidly achieved dominance. By the first half of 2025, so between January and June, an astonishing 92.8% of the H5N1 viruses sequenced in the Americas contained this new LPAI N1 segment.
93%. That's evolutionary takeover in real time.
It is. D1.1 essentially displaced the B and C genotypes that had dominated the epizootic landscape, and it did it within months. It proved it had a significant selective advantage.
So let's dig into the details of this new N1 segment, because it's not just new. It contains features that suggest a highly sophisticated change. What did the genomic analysis tell the researchers about the difference between the old Eurasian N1 and this new North American LPAI N1?
The analysis showed the North American LPAI N1 segment differed at 23 specific amino acid positions compared to the original Eurasian N1.
Okay, 23 positions. To put that in context for us, is that a lot?
It's a very significant number. It means there are 23 points where the basic structure of this surface protein has been altered. And these changes are not random. They're distributed across the key functional domains of the N1 protein.
And where are these key spots located? What do those locations tell us about the virus's strategy?
Well, we see changes in 2 main areas. First, there are 12 changes in the transmembrane and stalk domains.
And those are the parts that anchor the protein to the virus and influence its stability.
Exactly. But second, and this is highly relevant to human immunology, there are 11 changes on the globular head domain.
And the globular head is the primary target for our antibodies.
Yeah.
It's what our immune system tries to neutralize.
It is. Which brings us to this fascinating detail about something called the neuraminidase dark side.
The neuraminidase dark side, that sounds ominous. Where exactly are these changes on the globular head concentrated?
They are concentrated on the underside surface of that head, which is dramatically but accurately called the neuraminidase dark side, or NDS.
Okay, so if the globular head is like a mushroom cap,
The NDS is the underside of that cap, the part closest to the viral surface.
and why does changing the underside matter more than changing the top?
Because that underside, historically, is typically highly conserved across different influenza strains.
Meaning it usually doesn't mutate much.
Right. Since it's physically sort of shielded by the large sialic acid receptor molecule that the virus targets, it's not usually exposed to antibodies when the virus is active, so our immune system doesn't put a lot of pressure on it to mutate.
But something has changed.
Yes. Scientists have recently realized that this conserved NDS region contains effective targets for certain broad-spectrum, cross-reactive monoclonal antibodies.
The kind of advanced countermeasures we developed to fight many strains at once.
Exactly. So by introducing 11 amino acid changes right into this usually stable region, D1.1 is essentially mutating a back window that we thought was protected.
It's an adaptation that could potentially bypass our existing broadly reactive countermeasures. That's fascinating. It's not just adapting to the host.
It's adapting around our advanced medicine.
That is a profound risk.
It is. The authors point this out in their discussion. It's a major immunological concern. For years, we've relied on the fact that human exposure to seasonal H5N1 viruses,
Which contain an N1 segment that originally came from Eurasian swine.
right. We assumed that exposure provided some degree of cross-protection against H5N1 because the H5N1 that was circulating also had a Eurasian N1 segment.
They shared a common N1 ancestor.
Exactly. But now that D1.1 has swapped out that Eurasian N1 for a completely different North American LPAI-N1, that presumed cross-protection might be significantly compromised.
Or even lost entirely.
It could be. We are now in a race to urgently examine the new antigenic risk introduced by this single reassortment event. This is why this genotype is such a game changer. It forces us to reevaluate our immunological defenses from the ground up.
So we have a new genotype with a unique, aggressive, antigenic profile emerging in the Pacific. That alone is enough to cause concern. But now, let's talk about its behavior.
Its spread and its host range.
D1.1 did not waste any time.
That's the next layer of complexity here. D1.1 showed an unprecedented speed and pattern of spread. Unlike previous genotypes that might, spread linearly along one flyway, D1.1 dispersed rapidly across the entire North American continent.
And it moved predominantly west to east during that 2024-2025 winter period.
Yes, a complete reversal of the old pattern.
And the sources show that the scientists could actually quantify this speed. This is where that methodology, the Bayesian phylogeographic analysis, truly shines. They weren't just observing a fast spread.
No, they were calculating the viral velocity.
That analytical power is one of the clearest benefits of these sophisticated phylogenetic methods. The team didn't just track where the virus went.
No, they calculated its velocity using these complex metrics, including something called the relaxed random walk diffusion model.
Okay, that sounds incredibly technical. Can you give us some context for what that model is trying to measure and what numbers they produce for D1.1?
I'll try. Think of it like a GPS tracker for the virus, but one that tracks millions of movement possibilities all at once and then averages the speed and direction. The random walk part just acknowledges that viral movement is, random. It's driven by unpredictable bird migration and localized jumps.
And the relaxed part.
The relaxed part means they don't assume a constant speed. They allow the velocity to vary over time and space, which is much more realistic.
I see. So when they ran this model on the 17,500 sequences, what did they find?
They found that D1.1 showed the highest weighted diffusion coefficient among all the dominant genotypes they tracked, B3.2, B3.6, and C2.1. It was approximately 3,800 square kilometers per day.
3,800 square kilometers a day. That's a huge area for an organism to be expanding in every 24 hours.
It translates to an incredible speed. The calculation of the wave front velocity, so the speed at which the epidemic front was advancing during its initial invasion phase, was estimated at roughly 13,000 kilometers per year.
13,000 kilometers a year. To put that in perspective, how does that compare to the older dominant strains we were dealing with?
The difference is stark. The B3.2 lineage, the one that caused the mass marine mammal mortality, it moved at a comparable speed, so it was also highly efficient.
Okay.
But compared D1.1 to the C2.1 genotype, which was moving at only about 4,000 kilometers per year, or B3.6, which is moving at 2,500 kilometers per year.
So D1.1 is demonstrably a super spreader in the avian population. It's moving 3 to 5 times faster than some of its predecessors.
It is, and this rapid, widespread dispersal across all 4 flyways is precisely why it became the single dominant North American subtype in 2025.
Speed equals dominance in this evolutionary race.
Exactly. The speed and the geographic reach ensured that D1.1 was the lineage most likely to encounter new host populations and achieve more cross-species jumps. Which brings us to the other staggering element of D1.1.
Its host range.
Its host range.
So what stands out to you about how promiscuous this virus is in terms of the animals it can infect?
The host range is perhaps the most concerning trait of all, and it really speaks to the genotype's inherent generalist nature. The researchers categorized infected hosts into 7 defined types.
And D1.1?
D1.1 was the only genotype documented to successfully infect all 7.
All 7. What are they?
Wild bird, terrestrial mammal, marine mammal, domestic bird, cattle, domestic mammal other than cattle, so think domestic cats or goats and human.
All 7. It's not specializing at all. It's demonstrating this terrifying ability to adapt to virtually any environment it encounters.
And it's succeeding in those adaptations. It also had a broad host range within the wild bird population, detected in 12 different taxonomic orders, which equals the notorious B3.2 lineage.
So this generalist approach means it has the maximum opportunity for sustained circulation, for further adaptation, and for repeated spillover into human and domestic animal populations.
That's the danger.
So let's track that spillover history documented in the sources from late 2024 into 2025. This is where the human risk becomes undeniable and forces a global public health response.
The first documented human case that was sequenced as D1.1 occurred in November 2024. It involved a teenager who was hospitalized in British Columbia, Canada.
And there was a very worrying detail about that case.
There was. The patient reported no known animal exposure. And that implies either possible low-level environmental transmission or some other unknown routes of infection.
Which suggests the virus is circulating so widely and efficiently that direct contact with a visibly sick animal might not even be necessary anymore.
It's a very real possibility. This was followed a month later, in December 2024, by the first H5N1 fatality in the Americas, recorded in Louisiana.
And that was an adult with underlying conditions who was exposed to backyard poultry.
Yes. Then 2 months later, in early 2025, D1.1 was identified in dairy cattle herds in Nevada and Arizona. And that showed its ability to colonize these large-scale industrial settings, mirroring what B3.13 had done earlier.
And the human risk continued to manifest through those dairy operations.
It did. Following that Nevada cattle outbreak, a dairy worker there was infected in February 2025, and the exposure route was raw milk.
Fortunately, that case presented with mild symptoms, just conjunctivitis, similar to what was seen in workers infected with B3.13.
That's right. But the truly alarming case, maybe the single most chilling piece of data in the entire study, occurred shortly after that.
The fatality in Mexico.
Correct. In March 2025, a D1.1 infection in Durango, Mexico, resulted in the fatality of a young child.
And critically, the sources indicate the child had no underlying medical conditions, no travel history, and no known exposure to infected animals.
None. This fatality, happening in a young, previously healthy host, it just underscores the enhanced virulence and the risk posed by this particular genotype.
This brings us back to the molecular level. How is D1.1 achieving these cross-species jumps compared to that other dominant mammalian strain, B3.13? We need to talk about the viral machinery, specifically that PB2 segment.
Yes, the one that controls replication speed. This is a great point for comparison because it highlights the different evolutionary strategies the virus is employing.
So just to remind everyone, B3.13, the main cattle strain, it already had a fixed mutation, right? The PB2 M631L mutation.
That's right. And that mutation allows the virus to replicate more efficiently at the lower temperatures you find in mammalian hosts, especially in the human or bovine upper respiratory tract, which is cooler than a bird's internal temperature.
So because B3.13 already had this fixed, efficient mammalian adaptation, its spillovers typically relied on a bovine intermediary.
Yes, and it didn't need to rapidly acquire other mammalian adaptations. It was already pre-adapted, in a way.
But D1.1 is different. It's less reliant on that specific intermediary host.
Exactly. The D1.1 spillovers into humans, into domestic cats, into carnivores, they often correlate with direct exposure to wild birds. And in those subsequent mammalian infections, the virus shows a rapid de novo acquisition of key mammalian adaptation mutations.
De novo. So you're saying the virus is acquiring those necessary changes immediately after the jump into the mammal, not carrying a fixed advantage beforehand.
That's exactly right. The two most important mammalian adaptations here are PP2, E627K, and D701N.
And the E627K mutation is extremely well known for dramatically improving replication efficiency in mammalian cells, overriding that temperature restriction.
It's a critical factor in influenza's ability to jump species. And the sources indicate that in the 9 sequenced human D1.1 infections, that PB2 E627K mutation was found in 3 cases, and D701N was found in 1.
So this rapid post-spillover selection is what showcases D1.1's extraordinary adaptability.
The jump from bird to mammal is often a viable, efficient evolutionary path for D1.1, even without a specific intermediary host like cattle. It's adapting on the fly.
It speaks to this incredible viral flexibility. And if we, the scientific community, can track these adaptations, not just what the virus is, but what it is becoming in real time, we stand a chance of anticipating the next threat.
We do.
And that leads us perfectly into the journey of discovery itself. The authors of this paper, Crespo-Bolito, Trovam, and the rest of the team, they're using these massive data sets and complex computational tools to solve this very problem.
And their story is just as important as the virus's story.
The sheer effort of compiling 17,516 genome sequences and forcing them to yield a coherent narrative of time, place, and speed. It's just a massive undertaking.
It is.
So what is this core methodology, Bayesian phylogenetic, and why is it so essential for tracking a complex, rapidly evolving threat like D1.1?
Well, Bayesian phylogenetic represents a revolution in how we study infectious disease. It's a truly modern epidemiological tool that combines 3 key elements at the same time.
Okay, what are they?
Phylogeny, which is the evolutionary family tree of the virus. Time, which gives us the date of origin and evolution. And geography, showing us where it moved.
So instead of having 3 separate data sets, a family tree, a timeline, and a map, they're all merged into a single computational model.
Precisely. The research team uses these specialized software packages like BeastX, which analyzes the concatenated genome segments, all 8 pieces of the jigsaw puzzle linked together from those thousands and thousands of sequences.
And the software doesn't just show that A evolved into B.
No, it calculates the probability that A evolved into B at this specific time in this specific location. It's incredibly powerful.
It sounds like an incredibly computationally intensive process.
It's a Herculean effort. It requires high-performance computing clusters. They infer the evolutionary relationships, the time to the most recent common ancestor, that tight TMRCA window for D1.1 we discussed, and the spatial spread using these sophisticated statistical models.
Including that relaxed random walk diffusion model.
Yes, which is what allowed them to quantify that velocity, to confirm D1.1 emerged in mid-2024, and then calculate that it was the fastest spreading genotype. Without these tools, we would only see random clusters of outbreaks popping up across the continent.
But with them, we see the complete dynamic narrative of viral movement, which lets us predict where it might go next.
That's the goal.
This research effort really highlights the dedication of people like Crespo-Bolito and his colleagues. I mean, you can just imagine them compiling global data in real time, standardizing it with tools like GeneFlu, and then running these models to provide actionable intelligence. The journey they took wasn't just in the lab.
No, it was in compiling and cleaning massive, messy, real-world data sets.
And that's where the narrative of their journey encounters the practical dead ends and problems that need solving in the wider scientific community.
It's a huge part of the story. The research itself highlights serious limitations that hinder real-time tracking, and it proves that our surveillance infrastructure is lagging behind the virus's pace of evolution.
So let's start with what the authors identified as the most immediate problem for this kind of analysis: the lack of metadata.
This is such a crucial practical issue. Metadata is simply the contextual information about the sample, where and when it was collected.
Simple, but essential.
Incredibly, of the 1,722 D1.1 wild bird sequences that were deposited in the Sequence Read Archive, which is a massive public resource, less than 9% had complete collection dates.
Less than 9%.
That's right. Less than 9% had the day, the month, and the year, plus a precise sampling location, like the U.S. state, available in GenBank.
So that means over 90% of the sequence data is nearly useless for tracking speed or predicting spread.
It creates devastating blind spots. If you don't know exactly when and where a sample was collected, you can't accurately place it on the evolutionary timeline or the geographic map.
So the scientists have to make massive assumptions or just discard huge chunks of data.
They do. And it prevents them from tracking D1.1 with the higher precision that is so desperately needed to provide real-time public health insights. This data gap, it's not a fault of the computational scientists. It's a critical weakness in our field surveillance and data sharing pipelines.
And what about the problem of the background data? They're tracking reassortment, which means they need to know what native North American viruses the H5N1 is swapping segments with.
Right. And tracing that reassortment history is severely hampered by the lack of LPAI background sequences.
The low pathogenicity avian influenza viruses.
Exactly. You can think of the LPAI viruses as the ghost partners in this reassortment dance. Surveillance efforts, they naturally prioritize sequencing the highly pathogenic H5N1.
Of course.
But since reassortment happens with those North American LPAI, the native viruses in wild birds, we need far more sequencing of those LPAI viruses. Without that background context, it's difficult to fully resolve the phylogenies and definitively identify when and where those new swaps, like the N1 segment swap in D1.1, actually occurred.
And finally, there's the issue of language, or rather, nomenclature. If the global scientific community can't even agree on what to call the viruses, tracking them internationally becomes unnecessarily complicated.
That's it, exactly. The genoflu nomenclature, while it's vital for us here in the Americas for categorizing our specific reassortants A, B, C, D, it doesn't align with the systems used by European researchers.
So this lack of a unified global system creates a barrier to seamless international data sharing and coordination.
It does. We are dealing with a panzootic, a global animal pandemic that requires global cooperation. And that starts with having a unified terminology so everyone is talking about the same virus.
The sources conclude with a vital look at the global context and future concerns. For years, the global narrative has been that Eurasia is the source of the H5N1 threat and North America is the passive recipient.
Right.
But the authors suggest a major paradigm shift is underway.
They do, and they make a very compelling case. The authors explicitly connect the Americas as a new epicenter for H5N1 evolution. We are no longer just recipients.
We have a unique ecological landscape here that is driving rapid evolutionary change.
Yes. They cite two main reasons. First, the massive South American marine mammal populations, which are highly susceptible and provide an immense opportunity for sustained mammalian evolution. Second, the long-range industrial farming systems, particularly our dairy cattle, which sustain virus persistence in a novel mammalian host.
So the environment here is fertile ground for generating new, dangerous genotypes, and D1.1 is the perfect illustration of that accelerated evolution.
It is, and this trajectory remains highly uncertain. The sources point out that viral movement depends heavily on seasonal timing. For example, B3.2 spread so widely to South America because its peak activity in North American wild birds coincided perfectly with the southward migration window.
But D1.1 was different.
It was. D1.1, conversely, peaked during the winter of 2025, prior to those main southward migrations. So it remains unclear if D1.1 will peak again, or if an even newer genotype will replace it entirely before the next major migration season arrives.
And the sources conclude with a chilling warning sign that suggests we are already struggling to keep up with the pace of evolution here in the Americas.
That's the introduction of H5N5. A recent human fatality involving H5N5 was reported in Washington state in November of 2025.
Now, H5N5 was introduced into North America as early as 2023.
Right, but it was so sparsely sampled outside of the Atlantic flyway that it wasn't even included in this massive D1.1 study.
And the fact that a human fatality occurred in the Pacific flyway suggests that H5N5 has already established itself in wild birds there, likely for some time, without being adequately tracked by routine surveillance.
It underscores the author's central point. We are likely missing other emerging genotypes that could present immediate critical risks right now. The difference between missing 90% of the metadata and tracking everything accurately is the difference between having an adequate public health response and, well, flying blind.
And relatively small investments in standardized data pipelines and surveillance infrastructure could yield massive returns in tracking this zoonotic risk globally.
It really could.
So to summarize the D1.1 triple threat, it emerged unexpectedly and stealthily in the Pacific Flyway.
Right.
It achieved a major game-changing antigenic shift by acquiring the North American N1 segment, potentially bypassing existing human cross-protection.
Yes.
And it demonstrated the broadest mammalian host range and fastest spread of any genotype to date.
And this detailed phylogenetic analysis, the result of immense effort from the researchers in compiling and processing 17,500 genomes, provides the critical roadmap to understand those shifts.
It shows us that viral evolution isn't a slow, linear process.
No, it's a dynamic, geographically sensitive process driven by genomic swaps and powerful selective pressures in new hosts, like cattle. Understanding the precise timing and movement allows science to fight a fast-moving threat with equally fast, data-driven analysis.
Which brings us to our final provocative thought. For decades, the global focus has been on preventing Eurasian viruses from coming here and causing harm.
The whole model was based on that.
But the meticulous work by this team shows that the major evolutionary changes, the new and most dangerous risks, are now being generated right here in the Western Hemisphere, specifically through reassortment and spillover events like D1.1. So the central question going forward is, what risk does the D1.1 genotype, or its rapidly evolving descendants in the Americas, now present to the rest of the world?
It's a complete flip of the global public health script, and it's one that demands international attention and resources commensurate with the scale of the threat. And we must thank the authors for their detailed and immensely valuable work in tracking this complex evolutionary event.
The paper is Emergence of D1.1 Reassortant H5N1 Avian Influenza Viruses in North America by Alvin Crespo-Polito, Nidia Srivam, Alexander Maxiev, Guy Bayless, Simon Delacour, and Martha Sergelson.
And we also have to acknowledge all the contributing institutions who share this vital genomic data via USAID, NCBI virus, and other public platforms. This is the science, the diligent, computationally intensive tracking that keeps us ahead of the curve, even as the curve keeps accelerating.
Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other podcasts, responding to the content, and checking out our related articles at helioxpodcast.substack.com.